continue our discussion on nucleic acids. Now, what we learnt last time was how we have these specific bases, the purines and the pyrimidines, interact to form with double bonded hydrogen bonded structures, how they form complementary bases basically. Okay, so what we have here is, if we look at the nucleic acids, we know that they are now comprised of this pentose sugar, a phosphate and a nitrogen containing base, right? And we know that this pentose sugar can be of two kinds, either a deoxy kind or a ribose, a deoxyribose or a ribose, depending on the type of nucleic acid that you are considering. Now, obviously we have these two types, a deoxyribonucleic acid, where what is missing at the 2' position? The OH is missing at the 2' position, and we have the sugar and the phosphate, and the base families are the purines and the pyrimidines. And what do? They interact with hydrogen bonding, a purine and a pyrimidine, to form a basic two bases coming together in a hydrogen bonded network. Now, there is an additional factor that has to be considered here, that is the tautomerization possibility of the bases. Okay. Now, if you look at the adenine consideration here, what do you have here? You have an NH2 group. Okay. Now, what can happen to that NH2 group is this H can, you all know about keto-enol tautomerization. What happens in keto-enol tautomerization? What happens there? You have a C double bond O, and that is converted to an OH from a adjacent HCH2, right? So, you have a keto-enol tautomerization. Here we are having an amino type and an imino type, okay? But the basic idea is the same, where you are shifting this hydrogen in the case of adenine to the adjacent nitrogen. In the keto-enol tautomerization, what do? You have the H shifted from the carbon to the oxygen, right, where you have a keto-enol tautomerization. But what we are talking about here is an amino and imino case, which is possible in adenine and cytosine, right. So, we have the adenine structure. Now, where is this attached? This is attached to the, where is this attached? This is attached to the sugar ring, and what type of a bond is this? Beta N glycosidic bond. Okay. So, we have the adenine and the imino form. This is the amino form and this is the imino form. The same case with cytosine. We have an amino form and an imino form. Okay. So, we have now two other bases that we also have to look into. They are guanine and thymine. But what is the type of tautomerization here? It is a keto-enol type of tautomerization. Okay. So, when we are looking at the bond here, what do we have? We have the double bond O go to an OH. So, that is the difference in the type of tautomerization that you can see in A and C. So, what is the type of tautomerization you see here? It is an amino-imino type of tautomerization, where your hydrogen is shifted from the amine group to form an imine. Okay. So, we have this H shifted, this H shifted in cytosine. So, when we have A and C, we have amino-imino type of tautomerization, but when we are talking about guanine and thymine, we have keto-enol type of tautomerization, where the keto group here becomes OH. In guanine, in this purine, and in thymine, the pyrimidine, again we have the double bond O, the adjacent H goes to the keto C double bond O to form the enol. Okay. So, this is, you understand that since we are talking about the hydrogen bonding between the bases, this becomes an important factor. Okay. This is why we have to consider the tautomerization, because what you are doing is you are shifting the position of the hydrogen. Now, in shifting the position of the hydrogen, what are you doing? You are either disrupting a hydrogen bond or you are making a hydrogen bond feasible in a sense. Okay. So, if we look at the linkages, I will show you the hydrogen bond formation in a minute. But we considered the polymer formation in our last class, and what did we find? we find that this 3 prime end, what happens to the free OH? What does this free OH do? It goes and attacks a triphosphate, the alpha phosphate of the triphosphate, releasing the pyrophosphate and attaching the next nucleotide. What are we doing then? From these mononucleotides, we are building up our polynucleotide. Okay? So, we go from the 5 prime to the 3 prime end, and we will see how the other strand of DNA is the opposite of this. Okay? The 5 prime is at the bottom here and the 3 prime up there, and then we have the complementarity of the strands. That is what we are going to look at today. Now, in this, what do we have common here? I mentioned this last time. We have, what is this orientation that has been shown here? Is it a syn orientation or an anti orientation? It is a syn orientation. The anti orientation will have a rotation about which bond? About the beta N glycosidic bond. Remember, we mentioned about the types of flexibility, the types of angles of rotation that we can have, right? We can have a xy rotation about this beta N glycosidic bond that is going to render the base either in the syn orientation or in the anti orientation. And usually we have the anti orientation to prevent any steric clashes and also to assist in the hydrogen bonding in the complementary base of the other strand. So, the features that we are looking at here is the strand build up from the 5 prime to the 3 prime end, the addition of a mononucleotide in each case to form the polynucleotide. Now, what we know is each of these have a common backbone, the sugar phosphate backbone. So, usually when we represent it, we have a 5 prime end and a 3 prime end, okay? And this is the 3 prime 5 prime phosphodiester bond. What do we mean by that? That this is the 3 prime position and this is the 5 prime position. So, when we mention the phosphodiester bond, it is a 3 prime 5 prime phosphodiester bond. Is that clear? Okay, because what we are going to look at later is we are going to look at cleavages. Okay, how there are certain certain enzymes that will break only this bond. There are certain enzymes that will break only this bond. So, there is going to be specific, what type of enzymes do you expect the name, the nomenclature to be in this case? You are working on nucleic acids. So, what is the enzyme going to be? A nuclease, right? We have studied the mechanism of ribonuclease before. What did that work on? That worked on RNA, ribonucleic acid. So, when we are going to cleave or we are going to look at the cleavage of the nucleases, if it is going to cleave ribonucleic acid, then we will have a ribonuclease. If it is going to cleave a deoxyribonucleic acid, what are we going to have? A deoxyribonuclease, as simple as that. Okay. But what is the structure that I have here? Is it a DNA or an RNA? It is an RNA. Why? Because the OH is there, as simple as that. Okay. So, now when we have a representation, you know what the 3 prime 5 prime phosphodiester bond means. This is the phosphodiester bond, 3 prime 5 prime. If you have an addition, it is going to be added on this side, right? Just similar to how we had the polypeptide nomenclature. You had the amino terminal and you had the C terminal. Here you have what is called a 5 prime end and a 3 prime end. Now, usually you know that you have a sugar and you know that you have a phosphate. So, even though this is a certain representation that is easier to understand how the cleavage actually occurs, you usually do not even write this. You just write the bases. Like I mentioned last time, instead of the polypeptide sequence with the peptide bonds and so on and so forth, you do not write the peptide bonds, you just write the specific amino acids that are linked to one another, because you know they are all linked in the same fashion. The same goes for nucleic acids. You know that they are all linked by 3 prime 5 prime phosphodiester bonds, so there is no need to write the sugar or the phosphate. So, all you essentially do is simply list what is called the base sequence, instead of just like you would list an amino acid sequence. This is something that we looked at last time, where we have a guanine and a cytosine, we have a thymine and an adenine, okay? And we have in this case 3 hydrogen bonds possible. In this case we have 2 such hydrogen bonds possible. We also looked at the Hoogsteen base pairing, where we have a different face for the base pairing, and we also have in this case an, what is this? We have 2 purines linked together, where in the normal case, in a Watson-Crick case, you would always have a purine linked with a pyrimidine. Here again we have a purine linked with a pyrimidine, and if you notice, what we see is this 6-membered ring of adenine is involved in the Watson-Crick base pairing, whereas in the Hoogsteen pairing we have the one nitrogen of the 5-membered ring also take part in the hydrogen bonding between another base. Okay? So, this is what is called the Hoogsteen phase, and this is what we call the Watson-Crick phase, and this would be an example of an AU pair. Where would you see an AU pair? In RNA, because uracil is a base in RNA. So, when we are looking at a hydrogen bonding situation like this, we know we are looking at RNA, and we know we are not looking at a Watson-Crick base pairing, because you can straight away see that the base pairing is between the nitrogen of a 5-membered ring and a nitrogen of the amino group of the 6-membered ring. So, as soon as you see this, you know that this is a Hoogsteen base pair. Okay. Now, this is what I mentioned quite quickly last time, but it is extremely important in the 2 strands coming together. What do we have here? Here we have a Watson-Crick base pairing. Why? Because we have the 6-membered ring of the adenine of the purine involved in hydrogen bonding. Okay? So, this is Watson-Crick hydrogen bonding. We have the 2 hydrogen bonds between A and T. So, we are talking about DNA now. The distance, which is extremely important when we consider how the DNA structure is in its double helical conformation, the distance between the C1 primes of the purine and the pyrimidine is, well, it is actually 10.85 angstroms, but you could call it 11 angstroms. This distance is constant. It is also the same when you link G and C together. Now, that is extremely important if you consider the double helix, as I mentioned last time, because when you have the pairing of the ribbons of the double helix, they have to be equidistant from one another. So, we have the pairing come like this, and then we have a twisting of the helix, the double helix that it is called. What is going to happen is you are going to have a constant distance. So, what is that going to ensure? That is going to ensure a constant distance of the sugar phosphate backbone, right? Because you have a constant distance of the sugar phosphate backbone, how do you get that? It is because these 2 base pairings coming together are giving you that constant distance of 10.85 angstroms. Okay, and that is extremely important you understand in its structural aspects. So, what we have here is in the hydrogen bonding, where we have 2 hydrogen bonds for the A and T case, we have 11 angstroms. Again, when we have the hydrogen bonding for this purine pyrimidine set, we also have 11 angstroms. So, what we have is, in the DNA components, we have our 2 strands. What is this? This is the sugar, and this is, these circles are the phosphates. Okay? So, we have the sugar phosphate backbone, and sticking out from the sugars are all the different bases, and we have the complementary bases on the other side. And we know now that the distance between the C1 of this and the C1 of this is 11 angstroms. So, what do I have? I have a constant distance. So, I have a parallel orientation here. I have a parallel, absolutely parallel set of backbones. Is that clear? Okay. And why do you have this? Whether you have the AT set or the GC set, it is the same, because the distances are 11 angstroms and because you are linking a purine and a pyrimidine. You are linking one fused 6 and 5 membered ring, and you are linking it with another 6 membered ring, and the hydrogen bonds are between the A and the T, 2 hydrogen bonds, and 3 hydrogen bonds between the G and the C. So, this is what we have. This is what is usually called a ladder conformation, because it looks like a ladder. So, we have a ladder conformation, and the 5 prime end to the 3 prime end is what is actually synthesized. Okay? This is called the template strand. Okay? No, it's called the coding strand, the sense strand rather. This is called the anti-sense strand. It goes from the 3 prime to the 5 prime. Okay, so let me just reiterate that. We have a coding strand that goes from the 5 prime to the 3 prime. We have, this is also known as a sense strand, because that actually has the information. What information am I talking about here? What information does this DNA strand have? It has the information that is going to go from the DNA to the RNA, that is then going to decide what protein has to be made. Okay? So, this has the sense in it, basically, the first strand here. Then we are talking about the other strand, which is the anti-sense strand. The anti-sense strand, it goes from the 3 prime to the 5 prime, and this is also known as the template strand, because it provides the template for the mRNA, the messenger RNA, which we will see in a moment. Okay? So, if we go back to the slides here, we have the ladder conformation here, where we have one nucleotide specified. What does this nucleotide have? It has a sugar, it has a base, it has a phosphate. Okay? And when we consider the double helical structure, this is the double helical structure, where this set of parallel strands are twisting around one another. Okay? So, what we have is we have a constant distance here throughout and a twisting. So, it is called a double helical structure. We have two forms there twisting around one another. And what do we have? We have complementary base pairing, 2 hydrogen bonds for AT, 3 for GC. So, this is what it would look like. We have a sugar phosphate backbone and we have the bases. Now, when we look at this double helical structure, we see that there is a large gap here, this gap. There is a large gap here alternating, right? Because of the orientation or the way the structure is, we have these grooves. These are called grooves. This is what is called the major groove, okay? And this is called the minor groove. So, we have a major groove of DNA, we have a minor groove of DNA, and there are certain interactions between proteins, between other compounds that either set in the minor groove of DNA or they set in the major groove of DNA, depending upon what sort of interactions and obviously the sizes of the compounds or the molecules that we are talking about. So, if we look at the structure, we have a major groove of DNA and we have a minor groove of DNA. Okay. So, now I have just listed here the features of the Watson-Crick pairing, because it is going to help us in looking at the distances. We have 2 permitted hydrogen bonds between adenine and thymine, 3 permitted hydrogen bonds between cytosine and guanine. This is something we have seen. The dimensions of the 2 permitted base pairs are similar. The C1 prime to C1 prime distance is nearly identical in both cases. The beta glycosidic bond is attached on the same edge of the base pair, and even though we have the specific hydrogen bonds that are shown for the normal Watson and Crick pairing, you recall that there are other nitrogens also available. For example, the Hookstein pair looks at another nitrogen altogether, right? So, it means there are still other potential hydrogen bonding partners available. Now, this is important because later on these may be involved in interactions with proteins or interactions with other compounds. Okay? And another thing that is important is that the Watson-Crick base pair is a planar structure, and this is going to have certain implications in the stacking of the bases in the structure of DNA. Okay? So, these are the features. We have gone through all of these features, and we now know that the structure of DNA looks like this. Okay? So, the double helix, we have 2 nanometers between the strands. We have 1 complete turn, that is 3.4 nanometers, and the distance between the bases is 0.34 nanometers, and we have a major groove and a minor groove. Okay, so, these are the essential features of the double helix. Apart from the Watson-Crick base pairing, which is obviously extremely important in its structural aspects, we also have considerations of the major groove and the minor groove, what is called the pitch and what is called the distance between the 2 strands, which is constant because the base pairing, complementary base pairings are constant. Okay? So, these are the features that we have to consider. Another feature that we can look at here is the one, this part on the right. What groove is this? This is the major groove, and so this is the minor groove. Now, if you look at these are actually water molecules. Okay? So, there are other hydrogen bonding possibilities due to what is known as a water spine. Okay? The water spine that follows through the minor groove. Okay? So, we have a spiraling water spine that goes through the minor groove of DNA. Okay? The major groove actually is probably too large for this water molecule to sit in there, and this is a part that is also interacting with the other compounds and the other DNA protein interactions that occur. Okay? So, we have now how can we have these extra hydrogen bonds? What other units do we have? We have other heteroatoms present, right? We have phosphates, we have the phosphodiester bond present, right? That also has an oxygen. Then we have the nitrogen of the bases also, but the bases are within. So, we can also have what is called intercalation. Okay? Now, let me just show you where we can have intercalation. Okay. If we have the bases like this, you see how they are stacked on one another. Okay? So, we have like a stacking part here. Okay? So, when we have the stack of the DNA base pairs, we have them like this stacked one on the other. You can have intercalation. What does intercalation mean? Something that is going to set itself in between. Now, this is very important if you want to design any say drug that is going to cleave the DNA. Okay? Any drug that you want. Now, what is going to happen if you cleave the DNA? If you chop up this DNA, what is going to happen? It is not going to prepare the proper RNA, and that RNA will not be able to form the protein. So, essentially you are going 2 steps back into preventing a protein formation, right. That is a lot of research activities going on here, going on even in this department. What we have here is you have intercalating agents. When we speak of intercalating agents, we are talking of agents that are going to disrupt certain interactions. What happens when you have a protein? When you heat a protein, you add urea to the protein. What is happening to it? You are denaturing the protein. You are rendering it inactive. In this case, what you are doing is you are rendering the DNA inactive, in a sense that when you have these intercalating agents, it will prevent the proper stacking, it will prevent the proper interactions that are to be there, and it will then prevent the proper protein expression. Okay? Now, there are 3 types of DNA. The original Watson and Crick model, which just completed 50 years, it was first discovered in 1953, and the paper is available on the net, where you can look at the original paper of Watson and Crick that came out in Nature. You have bDNA, that is actually the most prominent form of DNA. It has 10.5 bases per turn. What are we talking about here? We are talking about a single turn of the double stranded helix that goes up, and there are 10.5 bases per turn. Just like we had a certain pitch of the alpha helix, it is similar to something like that. When we have the aDNA, this is formed when bDNA is chemically treated. It basically does not have those water molecules in the water spine. That is what aDNA is, and it has 11 bases per turn. The zDNA, as it is called, is a left-handed helix with 12 bases per turn, and it usually plays a role in gene expression. So, these are the 3 forms of DNA, and the most common by far is the bDNA. Okay. These are some of the features of the aDNA, bDNA and zDNA. We have a pitch. What is this pitch? What is a pitch? The distance covered by one rotation. So, the aDNA pitch is 2.8 nanometers, the bDNA is 3.4 nanometers, and the 4.5 nanometers for zDNA. The base pair repeats are 11 bases per turn, 10 bases per turn and 12 bases per turn. The twist per base pair, you realize that there is a slight twist as you have the base pair, like you would also have angle disposition for the alpha helix. These are the twists per base pair, and we have a slight base pair tilt, which is not very much in the bDNA, just 4 degrees. Okay? That is a slight base pair tilt. So, the 3 DNAs, we have the bDNA, the aDNA and the zDNA, and the most common structure that we will be considering is just the bDNA. Okay? That has about 10 base pairs per turn, 3.4 nanometers and a base pair tilt of 4 degrees. So, that the double helix of DNA is actually, well, it would not be a secondary structure, that is the structure of DNA, but there are other forms of DNA also. Studies have shown that the native intact form of DNA can be linear and circular. If we look at the double helix here, if it goes straight up and straight down, we would have a linear structure. Now, what happens is, if the 2 ends are covalently joined together, okay, So, if we chop this up, we are going to get a linear form, right. So, when we have a covalent joining of the two ends of a linear double helix, we obviously will get a circular helix. Okay. Now, the circular helix remains actually in these two forms. This is the tertiary structure of DNA. We have a relaxed form and we have a supercoiled form. Okay. Now, in the supercoiled form, there are certain terminologies that are used, something that you just probably need to know, so that we have a linking number, we have a twist, and we have a what is called a writhe. Okay, this is referred to as L, T, and W. What happens here is you have something like that. Okay, that is formed from this. You can even have this go even further. Okay, so you are basically going to have, now you have to remember that each of these is also a double stranded part. This that I have drawn here is actually this, right. It is the linear part of the DNA that has covalently linked the ends to form this. So, it's this is actually something like that, right, which you have joined together to form this circular DNA that has then further twisted itself. Writhe is like a wringing motion that you have when you suppose you're squeezing your clothes or something. Okay. So, you would have a link, a twist, and a writhe, but the reason why I am telling you this is when we consider the tertiary structure of DNA, see, as you combine this, you can get smaller and smaller parts, structural aspects of DNA. So, what you have is this is useful for the storage of DNA. You cannot have the linear strands all around, right. So, what happens is you have this supercoiled DNA that is actually the way you would have the DNA being stored, okay, because the DNA has to be stored. There has to be this DNA get to RNA, transfer its information to RNA, and this information has to be stored in the DNA. Okay. And because this linear strand does not remain as such, we have what is called supercoiled DNA. Okay. This is when you have DNA duplication. You have these two strands. What is this? What is this region? This is the major groove of DNA. This is the minor groove of DNA, right. When we have DNA duplication, part of the strands unwind, okay, and we have the ones in blue here are the daughter DNA molecules. Okay. So, what is happening here? You again have a similar strand form here. If this is A, what was it originally linked to in the DNA double helix? A T. When we have the new strand being formed, it will also be linked to a T. So, what do I have? I have a replication. Don't I have a replication? I am just doubling up the DNA that I have. So, it is a duplication. Okay? Well, duplication is probably the correct terminology that should be used. So, we have a duplication of the DNA. So, where I had one strand, now I have new strands, right. So, I have my daughter DNA molecules also formed from the parent DNA molecules. Okay. Now, RNA. Now, considering that the DNA structural aspects we always have as a double helix, in RNA it exists as single stranded molecules. These single stranded molecules usually do not take on an extended secondary structure like DNA in the form of a double helix. So, you understand that in this case there is no necessity for a complementary base pairing, right. We do not have to maintain a distance because we do not have to maintain that linear polymer, the parallel ladder like structure. In this case, the strands usually fold in a uniform periodic pattern, and we have several structural elements that are observed. We have what are called bulges and helices and so on and so forth, which we will see in a moment. But the basic differences here are that we do not have an extended linear double helical structure like DNA has. It usually is single stranded and it folds, and while it folds, it forms different types of structural elements. What are these? These are hairpin turns. Hairpin turns are loops in the chain that bring together complementary base pairs. So, when we are talking about RNA, actually we have a strand, a single strand. Okay? I may have an A here, a G here, a C here, a U here, another A here, and so on and so forth, a C here. So, what might happen in certain cases is we know that this G forms hydrogen bonding with C. Okay. So, in some cases we may have complementary base pairing. What is that going to result in? It is going to bring part of these two structures close to one another. So, you are going to have probably something shaped like that. Okay. So, it is not as regular as what you see in DNA at all. Okay. It is just parts, bits and pieces of the structure coming together into forming different types of structural elements that are either what are called hairpin turns, a right-handed double helix at times, and internal loops and bulges. These are very common in RNA structures, and usually they do not allow the formation of the double helical region of RNA, and this is because you do not have any complementary base pairing there. Okay? So, if you look at the structure, it is something like this. We have a bulge, we have an internal loop, and why do we have this internal loop? Because we do not have any complementary base pairing possible here. We can have helical segments here, we can have an unpaired loop at the tip, right. So, we have partial helicity here, and we have bulges, say it is probably such. So, now you understand in these cases there is a possibility for a Hooke's-Steen pair to occur, right, which is unlikely in the Watson-Crick base pairing for DNA. But here you can have a different type of base pairing because you do not have to have any regularity in the structure. For the DNA double helix to form, there has to be this constant distance of 11 angstroms between the C1 prime atoms in the double helix, but there is no such restriction here in the RNA, right. So, we have these single stranded RNA that just has all these helical segments, the loops and the bulges and so on and so forth. Okay. So, RNA is basically an adapter molecule that translates the information between, it translates from information in DNA and the protein, right. So, when we go from DNA to RNA, it is called transcription. RNA to protein is translation. Now, we have three types of RNA. This is something probably that you studied in school. You have messenger RNA. Messenger RNA is a transcription. The process of DNA going to RNA is called transcription, and this transcription of the information of DNA, and it carries the information in a temporary format from the DNA. Now, how does this occur? If we look at the previous thing that I was showing you here, this coding strand, suppose we have this from 5 prime to 3 prime, the antisense strand, which is the template strand, which is used by mRNA, is the 3 prime to 5 prime strand. What happens in this case, say our coding strand is A, T, G, G, A, or something like that. Then what is the antisense strand going to be? What am I going to have here? T, A, C, C, T, T, right? So, this is my sense strand that goes from 5 prime to 3 prime. This is my antisense strand that is 3 prime to 5 prime. Then I have my mRNA, which actually is linked to the antisense strand. So, what is the code for the mRNA going to be? What is it going to be? Because it is going to be complementary to this now. So, what am I going to get? A, then U, G, G, A, right? Why U? Because now I am speaking of mRNA. So, now when this is going to form the next, so but this information it has taken from this, right? You understand that. This is the sense strand, the coding one. This is the template. This is the mRNA. Okay? That is what it does in transcripting the information from the DNA. So, it has taken the information from the DNA now. Okay. We have ribosomal RNA. This actually forms the core of a large molecular machine which makes the proteins. Okay? It has a huge number of proteins. Not all of the structures of this have been solved yet. Okay? And remember, you looked at the RNA structure right now, where you had part, what did you have? You had part helices, part bulges, and so on and so forth. Well, this looks a lot more complicated when we look at the ribosomal RNA. You will have some bits and pieces like this, and then you have loops like this. If you look at a picture of, I couldn't get a proper picture, so it will look like this something, and they're huge, okay? They have associated with them proteins, and these are ribosomal RNAs are actually formed in the ribosomes, where they are used to make the proteins. Okay? We won't see actually the process of making the proteins, but it's extremely interesting. And if you go to the internet and you type in protein animation, okay, just animation of the synthesis of proteins, you can see how the mRNA and the rRNA and the tRNA, which we will speak about in a moment, all work in tandem to get you the peptide, polypeptide chain. The transfer RNA is the smallest type of RNA that has just 64 to 93 nucleotides. Okay? It has what is called a clover leaf structure, which I will show you in a moment, and it is folded up into an L-shaped tertiary structure. We have attached to each tRNA a specific amino acid. Okay? And this amino acid is required for the protein chain to grow. Okay? The information present in the tRNA will tell us from the genetic code which amino acid it is supposed to bring to attach to the polypeptide chain. I will show you that in a moment. So, what we have here is we have the smallest type of RNA, the tRNA. It has its classic clover leaf structure. Okay. So, this is what is called the classic clover leaf structure. Okay. This looks like this clover leaf has 1, 2, 3, looks like that. Now, what do we see here? Specific complementary base pairing, okay, but we have these bulges at the two ends. This is what is called the anticodon. Now, what happens here in the anticodon is this is the part that links on with the RNA to get the information as to which polypeptide chain or which amino acid has to be brought. Okay. Now, what I want to show you is, let me just go back here once. This is what is called the genetic code. Okay. Now, in the genetic code, I will just show you what it means. We have something like this. This is the first base pair. Let me just get this right here. We have T, C, A and G. Here we have say T. Here we have again T, C, A, G. Here we have C. Again T, C, A, G. This is something that was found by Hargobind Khorana. Okay. Now, what we have here is, this is the first, the second and the third. Okay. So, what is the first? T, T, T. So, we have T, T, T. The second one will be T, T, C. T, T, C. Third one is going to be T, T, A, T, T, G. Okay. In this case, we have T, C, T, because the first one is T. This is the first one, this is the second one, and this is the third position. So, we have T, C, T. The next one is going to be T, tell me, C, C. This one T, C, A, T, C, G, and so on and so forth. Okay. So, this is what your genetic code is. So, this is CTT, CTC, CTA, CTG. Now, each of these code for an amino acid. We have triplet codons. These are triplet codons. Okay? The triplet codons will code for an amino acid. These two code for phenylalanine. I probably should write this in a different color so that you can see it better. These code for phenylalanine. These code for leucine. This codes for tyrosine. This codes for tyrosine. This is actually a terminal codon. This is also a terminal. No, not this one. This is serine. Sorry. All of these are serine. The A ones are your terminal codons. So, you have TAT, TAC, TAA, TAG. And what do we, this is where we have tyrosine, tyrosine, terminal, terminal. So, you understand how many codes am I going to get like this? 64. They will code for 20 amino acids, right? We have an initiation codon and we have a terminal codon. The terminal codon, okay. So, this is how, if we go back to what we have here, this is what we have. See, if you can see this, then we have You get this as I've just got this from the internet. You have this coding for phenylalanine, this for leucine, and so on and so forth. Now, the interesting thing here is that you see for this case the third base actually does not matter. Why does the third base not matter? If you look at the leucine codes, just look at them. The CTT, CTC, CTA, CTG all give you leucine, right? So, it means that this base may be something else. And where is this base? This base is in our part here. This is the triplet codon. It's the anticodon for that triplet that is for the specific amino acid. What did we see in the genetic code? We had a triplet that corresponds to an amino acid. That amino acid is what is used for your polypeptide chain, or rather your protein synthesis. Okay? So, if we just summarize here in a comparison of the structures, we have the sugar. For DNA we have deoxyribose and we have ribose for RNA. The bases are AGTC and here AGUC. The strands are double stranded with base pairing. Here RNA is single stranded. This forms a helix, this does not. Okay? We will stop here for this class. Thank you. Our third lecture on nucleic acids is going to speak about the stability of DNA, okay, and what are the certain factors that are going to lead to the denaturation of DNA or the renaturation of DNA, and how that is extremely important for designing of drugs for the cleaving of DNA, as I mentioned in the last class, where we want to consider that the protein synthesis is inhibited or stopped. Okay. If we go back to look at the double helical structure of DNA, we have our sugar phosphate backbone and we have the specific bases linked together. Now, apart from the hydrogen bonding present here, we are going to have certain other interactions that are going to be responsible for the double helix to maintain its structure in solution. Now, the forces that actually result in the DNA structure are electrostatic forces that are mainly repulsion forces. Now, can you guess why we would call them repulsive forces? Why would the electrostatic forces in the case of DNA or the structure of DNA that I showed on the previous slide be called repulsive? What do we have in the backbone? We have sugar phosphate. What is the charge on these phosphates? They are all negative. So, the strands would tend to be as far away as possible, right, which is going to result in an electrostatic force that is going to be basically a repulsion. We have hydrophobic forces, we have hydrogen bonds obviously between the bases and also possible between the other heteroatoms present in the bases with the water that is around, and we have stacking interactions between the bases. Between the bases at different levels. Now, considering the properties of the Watson-Crick base pairing, the base pairings are planar. Okay? So, in the resulting planarity that we have between the GC and the AT pairs, we are going to see a stacking interaction that again can be disrupted with any agent that penetrates this region between the bases. So, we have these 4 types of forces that actually determine the DNA structure. Now, the base pairs are usually found in the interior of the helix, okay, which is going to result in the stacking interactions between the bases. The charged and hydrophilic sugar phosphate backbone are on the exterior, which results in the electrostatic repulsion of the strands. The phosphates from the 2 strands would obviously be as far away as possible. Why? Because they are negatively charged and they would reduce the electrostatic repulsion if they would be as far apart as possible. Some of this is neutralized by polyamines. Why would they be neutralized by polyamines or magnesium? Because they would be positively charged, they would counteract the negative charge of the phosphate and result in stabilization. Usually we have magnesium and polyamines that would result in the stability of DNA. So there are these. So DNA actually is quite stable, and in mild acid solutions at pH 4, in this case we have a different kind of hydrolysis whereby the purine bases themselves are hydrolyzed. Okay? Since the OH is absent at the 2' position, unlike RNA, what happens in this case, the beta glycosidic bond to the purine bases are hydrolyzed, and we also have protonation of the purine bases. Now what is going to happen if you are going to have protonation of the purine bases at the acidic solution? You are going to disrupt the hydrogen bonding, right? As a result of which you are going to have this hydrolysis which leaves your purines protonated, that is going to act as a good leaving group, and you are going to have an isomerization of the depurinated sugar. This is the hydrolyzed sugar, where you are going to have, what is this 5' and what does this signify? What does this 5' and 3' end signify? It signifies that this is part of a DNA strand, right? You do not have the OH here, so this is part of the DNA strand, and this occurs under mild acidic conditions where there is a protonation of the purine is lost, and what happens is this sugar now that is on the left hand side, which is depurinated, isomerizes to an open chain form. So what have you done in mild acidic conditions? You have not only depurinated your DNA, you have also opened up the sugar, right? So that could basically be a problem. What happens in RNA? This is RNA now, right? So in the RNA, what is this doing now? This is in RNA, what we have here is